Hi, in this video we look at the cochlea and the vestibule. So this is the bony part of the cochlea within which it has a membranous labyrinth which is called the cochlear duct. And the cochlear duct is connected to the vestibular membranous part which is called the saccule and the utricles. Now here the utricle is connected to the semicircular ducts and which has a swelling at the beginning called the ampulla, and this semicircular duct is protected or is within the canal called the semicircular canal, which is bony structure. So here are the three ampullas and they have the semicircular ducts which are within the semicircular canals, and they are the lateral, the posterior and anterior semicircular canals within which it has the ducts. Okay, so here is the saccule, the utricle, the ampulla. If we look at the cochlear part which has the different areas, here is the scala vestibuli, and then here is the scala media, and then it has the scala tympani. So these different parts has the different fluid within it, right? And it's fluid-filled cavities. If we untwist the cochlea and show it outside here as simple tube like this one, so here is within it is the cochlear duct, and here is the scala vestibuli, here is the scala media, and here's the scala tympani. Within the scala media, there is a fluid called the endolymph, right? The inner ear is full of fluid, while the middle ear is not. It's full of air. So here, this endolymph, it flows in all the membranous labyrinth, which are the saccule, the utricle, the ampulla, the semicircular ducts, right? And here is the perilymph. And the perilymph is in the scala vestibuli, and it flows around all these other membranous labyrinth. Within the bony structure, it flows, okay? Now, let's look at the special structures within these systems. First, we look at the organ of Corti. The organ of Corti is in the cochlea, and it helps you to hear the sounds in the environment. And then in the saccule and utricles, there is another structure called the maculae. And then in the ampulla, you have the structure called the cristae. And the cristae, there are three ampullas which contains the different cristae. And if we look at one cell of the cristae, it has a special sensory epithelial cells. And these cells have projection at top called the cilia. And these cilia, they're covered by another structure called cupula. In cristae, it's covered by cupula, right? Here, this is the cupula. And if we look at maculae, it is in the saccule. In the saccule, it's on the wall, which helps you to detect the balance of the head up when you're going up and down. And in the utricle, it's on the floor, which helps you when you move your head forward, back, and the sides, right? So the maculae and the cristae, they are for the balance. And if we draw here one cell of maculae, right, it's for the balance, the cristae and the maculae, they are for the balance. If we draw one cell here for maculae and show the sensory epithelial cell of maculae, it has, again, the cilia on top, the projections is the cilia. And in this case, these cilia, they are covered by a jelly-like material. If I show here in this diagram as the example, so here are the different maculae special sensory epithelial cells with the projections. Let's imagine they are the cilia, and on top it has a jelly-like material in red. Okay, so this is jelly-like material. Within the jelly-like material, it has calcium carbonate stones called these ones, called the otoconia. Now, these otoconia, when you move your head, they can slide over these cilia, and bending the cilia, either they will be stimulated or inhibited, okay? So we look at another structure here, and it is the organ of Corti. And if we make a section in the cochlea, right, and then draw it here outside, let's say we do the section at this part and draw it outside here. So here are the bony structure of the cochlea, and here's the bony small triangle specule within which there is the spiral ganglia. Okay, so here are the three structures that we will show just now. Let's imagine this is the section of the cochlea, and here is the basilar membrane. Here is the basilar membrane, and then here is the Reisner membrane. The Reisner membrane here, and it has the organ of Corti within the. Okay, so let's look at these areas. Here is the scala vestibuli, the scala media, and the scala tympani. Within the scala media, you have the endolymph, and there you have the organ of Corti in red here. Okay, here is the organ of Corti. So if we look at the organ of Corti and magnify it, so here is the triangle shaped bony specule, and here is the basilar membrane, and the basilar membrane has on top the supporting cell layer, and then there is a tunnel. Okay, and this tunnel is between the inner and the outer cells of the organ of Corti, right? So this is the inner tunnel, right? And on the inner side, it has the cells called the inner cells, and they make a row. Okay, so this is the inner cells. And on the outer side, there is 3 rows of the outer cells. So here are the 3 different rows of the outer cells. Now, on top of the outer cells, it has a membrane. Now, these cells are called the hair cells. They have a hairy-like projection. Now, this membrane holds this hairy-like projection of these cells. So here is a membrane called the membrane tectoria, right? If we look at here, it's the inner cells, and here is the outer cells. The inner and the outer cells, they are called the hair cells, right? And if we draw one hair cells outside here, it has the hair-like projections, which are held together by the membrane tectoria, right? So now, if we look at all these, this is the organ of Corti. If we look at all these different cells, they have the potassium channel. Let's take one example here. It doesn't matter which cell, but here is one cell, and it has the potassium channel in the projections, right? In the cilia or the hair-like projection. Here they have the potassium channels. These all cells have potassium channels. Now, depending on the potassium channel, when it opens, the cell will be inhibited. When it is closed, the cell will be stimulated. Okay, how? When you move your head, all these stones, they slide, or the bending of cilia or the hair-like projection occurs, there will be either stimulation of these cells or inhibition. Let's say we bend these cilia to this side, right? And here is the potassium channel, and it is closed in this case. And when it is closed, the potassium is a cation. It is a positive charge. When the positive charge is not leaking out, the cell will be depolarized, and it will be more positive, and there will be action potential, right? And means it's stimulated. But if these cilia, they are bent toward this side, and let's say the potassium channel is opened in this case, and there is a leakage of, or the potassium ions will leak, which are the cations, that will make the cell more negative, means hyperpolarized, and that means the inhibition of this cell, right? This is how these cells work. Let's say one hair cell is stimulated, the action potential will be taken by the nerve fibers, and there is the spiral ganglion in the bony specule here, and these peripheral fiber will come, and then the central fiber will go to the CNS through the vestibular, the cochlear nerve. Okay, so if we look at the cochlea and untwist or uncoil the cochlea here, here is the oval window, here is the promontory, this is the medial wall, and here is the round window. And if we untwist the cochlea and show it like this structure here, so here's the cochlear duct, okay, And if we show the different ossicular bones here, here's the stapes, and then we have the incus, and then we have the milius, and the milius attached to the tympanic membrane. Here is the tympanic membrane, okay? So then we label these areas as well. So here is the scala vestibuli, here's the scala tympani, here's the scala media, right? Here's the basilar membrane, okay? This is the basilar membrane. Now the sound waves will vibrate the tympanic membrane, and the tympanic membrane and the ossicular chain will take the vibration to the scala vestibuli. From the scala vestibuli, it will transmit to the scala tympani, where it will vibrate the membrane, the basilar membrane, and that will stimulate rubbing the hair-like projection or bending the hair-like projection against the membrane tectoria, and that's how the action potential will go through the nerves to the spiral ganglion, and then through the cochlear nerve to the CNS, means the brain stem or the cortex, okay? So in this case, if they are stimulated, the fiber will go through the nerve fibers to the vestibular ganglion, and from the vestibular ganglion, it will go to the from the vestibular ganglion through the vestibular nerve to the CNS, means the cortex or the brain stem, wherever they have the nuclei. So this is all about the vestibulocochlear system.